Hello students, today we will discuss about our new topic, that is bacteria and role in preservation and spoilage. Then we will discuss about the general bacteriology. First of all, we will discuss about the definition of bacteria. That is, bacteria are unicellular microscopic chlorophyll-less organisms belonging to plant kingdom, which reproduce by the binary fission. Then we will see the structure of the bacteria. That is, it is the section of a bacterial cell. Here you can see the cell wall of the bacteria, capsule, enfolding of plasma membrane, DNA coiled into nucleoid, basal body, flagellum, ribosomes are there, cytoplasm, plasma membrane, we can see the pili, cytoplasmic included. Okay, this is the section of a bacterial cell. Then the size of bacteria. Bacteria are so minute that they are visible with aid of microscope. Under microscope only we can see the bacteria. Bacteria are 0.5 to 1 micron in width and 2 to 5 micron in the length. Then about flagella of the bacteria. These are thin hair-like appendages. They arise from granular bodies which are present just beneath the cell wall. Flagella help in motility of the bacteria. Flagella is the main organ which help in the motility of the bacteria. Then third one is the pili. These are the filamentous structures. They are short but more in numbers than flagella. Pili are present in both motile as well as non-motile bacteria. They help in attachment of bacterial cell to the substratum. Then fourth one is the capsule. Some bacterial cells are enveloped in a thin slimy layer of polysaccharide. The capsule protects bacterial cell and also functions as a reservoir of food. Then fifth one is the cell wall. Cell wall is rigid. It is 10 to 25 millimicrons in thickness. It is made up of substances like diaminopalmetic acid, mamic acid, aminosugars and lipids. Then sixth one is the plasma membrane. It is present inside of cell wall. Plasma membrane contains respiratory pigment. It is responsible for respiration of bacterial cell. Then seventh one is the cytoplasm. Inside the plasma membrane there is cytoplasm. It is formed from water and organic matter and inorganic matter like calcium, chloride, magnesium, phosphorus, potassium and sulfur. Cytoplasm contains lipid protein and glycogen and ribosomes. Then the nucleus. There is no localized nucleus. It is located anywhere in the cytoplasm. It contains only DNA. RNA is absent. Then we will discuss about the classification of bacteria. First of all, cocci. Cocci, you can see the diagram of cocci here. The only one is the coccus. If it is present in the chain of which is in many numbers, that is streptococci. If coccus are 2, that is diplococci. Then staphylococci, tetrad, and then sarcina. These are the examples of the cocci. Then here we can see the spherical or coccus bacteria, that is monococcus, diplococcus, staphylococcus, tetracoccus, sarcina, streptococcus. Then rod or bacillus, that is bacillus, cocobacillus, palisades, diplobacillus, spore former, and streptobacillus. And curved shape of bacteria, these are the Vibrius, Spirilla and Spirochetes. Then bacilli, these are rod shaped cocci and spirilla. So we have seen the classification of the bacteria according to their shape and size. Then classification of bacteria according to staining. According to staining, Graham, a Danish scientist, distinguished the bacteria by differential staining. Bacteria stained with gentian violet, treated with iodine and then washed with methyl alcohol, and then they are counter stained by safranin. After observation, some bacteria retained main stain are called as the gram positive bacteria, and those lose main stain are called as the gram negative bacteria. Then we will see the classification of bacteria according to the nutrition. That is autotrophic bacteria. The bacteria synthesize their food material from simple substances. That bacteria we call as the autotrophic bacteria. They can synthesize their own food with the help of simple substances. They are again classified as chemosynthetic, means the bacteria build up their body substances from the inorganic matter. They get energy by chemical reaction. Then second one is the photosynthetic bacteria. They absorb energy from the light. In that heterotrophic, they are unable to synthesize their own food. They are again classified as parasite. They obtain their food from the living organism. They are of two types, that is endoparasite and ectoparasite. Then next one is the saprophytes. They obtain their food material from decaying organic substances that we call as the saprophytes. Then classification of bacteria according to the respiration. First one is the aerobic bacteria. Bacteria which live only in the presence of oxygen, that we call as the aerobic bacteria. And anaerobic bacteria are the bacteria which live only in absence of oxygen. These are the two types of bacteria according to the respiration. Then next one is the classification of bacteria according to the temperature. First one is the psychrophilic bacteria or psychrotropic bacteria, means the bacteria which can grow at refrigeration temperature, that is 5 to 7 degrees centigrade. And second one is the mesophilic bacteria, the bacteria which can grow at the temperature 20 to 40 degrees centigrade. And third one is the thermophilic bacteria, the bacteria which can grow at the temperature above 50. So they are heat loving bacteria. And next one is the thermodiuric bacteria which can grow above the 80 degrees centigrade temperature. Then we will discuss about the growth of bacteria. The growth of bacteria refers to an increase in number. The growth stages. The growth phases are initial stationary phase. Second one is the lag phase, means phase of adjustment. Third one is the log phase, phase of accelerated growth. And fourth one is the maximum stationary phase. And fifth one is the phase of accelerated death. The factors affecting growth of bacteria. Now we will discuss one by one. The first factor is the food supply. Every living cell requires a source of energy to maintain its life process and elements for the formation of its cell substances. Milk and milk products furnish all the food requirements of microorganisms. Okay, every living cell requires a source of energy. For what? To maintain its life process and also the elements for the formation of its cell substances. And milk products are the best sources for all these requirements of a bacteria. So they contain water, fat, protein, lactose, minerals and vitamin. The milk contain water, fat, protein, lactose, minerals and vitamins. So the moisture. Microorganisms are taking their food from aqueous solution and hence water is an absolute requirement. Microorganisms find the optimum condition for growth when the foodstuff and atmosphere saturated with moisture. Concentration of water in nutrient material above 30 is good for microorganisms. If there is percentage of moisture or water in the food is above 30% in that food, there will be growth of bacteria because this much of moisture is sufficient for the good growth of the microorganisms. Then next one is the atmosphere. Certain bacteria grow only in the presence of oxygen in the atmosphere while others are growing in absence of the oxygen. Some bacteria grow only in the presence of oxygen and some other bacteria, these are growing in the absence of oxygen. In milk, plenty of organisms can grow either in presence or absence of the oxygen. Then we will discuss about the chemical and physical environment. Most of the bacteria require medium which is nearly neutral in reaction, means pH 7. If acidity in the medium is increased either by action of microorganisms or the addition of acids, growth of bacteria is restricted. If acidity of the medium is increased, the growth of bacteria is reduced. For example, growth of ordinary souring bacteria in milk is reduced when acidity of milk reaches up to 1%. If acidity of milk reaches up to 1%, then the ordinary souring bacteria number will decrease or reduced. The next one are the preservatives. Certain chemical substances either check the growth or destroy the bacteria. Such substances are called as the preservative. For example, formalin, benzoic acid, boric acid, borax, salicylic acid, hydrogen peroxide, these are the examples of preservatives. The next one is the concentration of nutrient solution. High sucrose or salt content in products check the growth of bacteria. Osmotic pressure may develop condition by which growth of microorganisms is checked in nutrient. Then light. Most of the bacteria grow best in the dark. Light is harmful for the growth of the bacteria. Bright light tendency to check the growth of microorganism. Direct sunlight, ultraviolet rays are destructive to the bacteria. Then temperature. Each species of microorganism has minimum, maximum and optimum temperature of growth. The effect of temperature upon microorganism may be considered under three headings. Low temperature, that is below 50 degree Fahrenheit, that is 10 degree centigrade. Medium temperature 50 to 110 degree Fahrenheit, that is 10 to 43 degree centigrade. High temperature, that is above 110 degree Fahrenheit and above 43 degree centigrade. Then we will discuss about the reproduction in bacteria by binary fission. Here you can see in this diagram the plasma membrane, cell wall and DNA. The DNA duplicates and cell begins to divide into two. That is, two daughter cells are separated. Then many bacteria reproduce by the binary fission. The nuclear body of the cell divides and wall is developed from periphery towards center, separating two nuclear bodies. The next is the budding. Budding means In this case, the small bud formation from a parent cell and daughter cell finally gets separated from the parent cell. There is small bud formation from the parent cell and that bud is separated from the parent cell as a daughter cell. Here we can see in the diagram. Some bacteria develop outgrowth called as the buds. These buds separate and grow as a new cell. And then the next one is the conidia. The actinomycetes reproduce by means of conidia. The conidia are spherical or rod shaped in the structure. Under favorable conditions, conidia germinate to form new filaments. The picture of endospore. Here you can see the endospore, parent cells, the core cell you can see, exosporium, spore coat, plasma membrane, cortex, cytoplasm. So from that the new bacterium is formed from the endospore. Species of Bacillus and Clostridium produce endospores which are rich in calcium, are resistant to the heat, chemicals and desiccation, and can survive unfavorable condition. Okay, generally a single spore is formed per cell. On the onset of favorable conditions, the endospore germinates to form a single cell. This is a reproduction without multiplication. Then we will see the sexual reproduction. Some bacteria may produce by sexual reproduction. The procedure is as follows. The formation of specific pair of donor or recipient is there. Joining of donor and recipient cells by the sex pili. Formation of conjugation bridge. Transfer of chromosomes from donor to recipient cell through conjugation bridge. Integration of transferred chromosomes in the recipient's chromosome. So this is all about the sexual reproduction.